100 different people and then get genome data for all of them and then see what mutations are there, what is the set of mutations that are occurring, or what is the frequency of each mutation, right? And is the mutation causing any changes in them? So more than epidemiology, you can say population genetics here. In this case, epidemiology is more about spread of diseases. Population genetics dictates that some populations are more susceptible to some diseases. How do you know that? You know that through omics studies only. Population studies also dictate that there's an effect of environment on this that can also be understood from omics. So in order to, wrangle so much data, all this data, imagine what I'm a bioinformatician. And believe me, when I was starting out, it took me 3 months to get to understand one data set. Because, can anyone tell me what this data would look like? I'm talking about omics data since so much time. Does anybody know what it looks like? Okay. ATGC. Okay. Okay, I noticed that we are at 7. If the organizers are okay, I can go on for another maybe 15-20 minutes. Is that fine?
Yes, ma'am, we can go for another 15 minutes.
Okay,
I
great.
think we can, you can let them know about how AI is mostly used in this. Yeah.
Yeah, the next thing is on AI. So I'll quickly do that and then we can take any questions we have. Okay. So yeah, thanks. So what I'm trying to say is that, when I was doing this, right, I was given a single cell data set. That means I was given a huge Excel table. Imagine that, giving a biologist an Excel table and saying all the rows are genes and all the columns are cells. So that means for each cell, I have about 15,000 human genes and the values of each genes. That means the number of that particular gene. Let's say TP53. Okay, in cell number 1, there are 50 TP53 genes that are found in the cell physically. Similarly, some other gene has 20. And this changes for every cell. And I have to understand what different cell populations are there based on this. That means which cells have more TP53, which cells have more P10. So imagine the amount of, it took me 3 months to get through this data with a lot of help. And even today, even seasoned bioinformaticians and biologists struggle with dealing with these many different kinds of data. You have bulk, you have single cell, you have spatial, so many different kinds of data, proteomics, all different. How will you combine them? We discussed about why multi-omics is important, right? It's not, you can't isolate one particular thing and analyze it. Or if you're doing multiple people, analyzing multiple people, how do you do that? Right? And that is when, and 80% of bioinformaticians' time goes in dealing with this data than actually solving a problem. So AI comes in here. AI will give you speed, scale, and the ability to connect those layers into actionable insight. There's no point making huge statistical analysis if you're not getting an insight out of it. So AI is known for taking huge amounts of data, applying statistics on it, and giving out insights from it. So I'll quickly cover 3 cases of where AI can solve. One of them is called graph neural networks. This has been used for repurposing drugs in many important diseases. So what they actually represent here is, this is called network biology, and what they are trying to represent in this graph is that in a disease, there are a lot of proteins that are responsible. There are drugs that are known in literature. There are genes, and then there are drug targets. What we all know already is that one drug can act on 2 different diseases because it may be targeting the same protein in 2 different diseases, right? Similarly, one disease can have multiple drugs that are effective. So what we do is we take all this information from literature and make a huge graph where each drug, let me show you the next slide. Right. So as you can see here, each drug is related to a gene. It is also related to a disease. And drug is also related to a side effect. Similarly, each gene is related to a disease, a drug, and a pathway. Right? So if I make a huge graph of this, let's say just for one disease, I'll have multiple, let's say 5 drugs. Each drug has its side effect. Each drug also has some other disease that it is helping treat. And then there are multiple genes for each disease. So what we do here, this is called a knowledge graph. What we do here is we give this model to neural networks and other forms of different kinds of AI models. What they do is they learn this pattern. How do they learn this? It's called masking. So they hide this. They first learn everything, then they hide this and say predict a new gene. So what we do here is drug repurposing. And now when a new disease comes in, it will be there in this huge knowledge graph somewhere, right? Or let's say I found out that for some cancer, let's say a new gene has been known to be active via bulk transcriptomics. Now I want to see what drugs can I give. So what you'll do, you'll just put a gene into this network and see where it is connected. So the whole point of GNNs and drug repurposing is use all the literature that is available right now, give it to an AI model to learn patterns and relationships between these. And now when you put a new element into it, you can easily trace the network back to what kind of drug you want or what disease you want. This involves a lot of coding, but we have technologies that can help you automate this as well. The next thing is one of the most advanced methods that we have right now, also very important, is called foundation models. So now, see, people who are trying to solve diseases, treat diseases, solve problems, understand new disease mechanisms are mostly biologists, bioinformaticians, clinicians, and doctors. So for all of these people, it's very difficult to leave everything, sit on a system, and then try to wrap your head around all of this, right? What we can do though is like I'm sure you all use ChatGPT at some point. So what ChatGPT basically does is you give it huge amounts of literature and you ask any question, now it will be able to answer your question. How is it doing that, right? The thing is, ChatGPT learns from all kinds of books. Doesn't mean that your query is necessarily there in that book. Even if you ask a new question, it'll be able to answer because it has learned not the books, but the patterns and the language that occurs in that books. Similarly, imagine a cell is one book and all the genes in the cell are the words. So if you can see here on the left hand side, this is how these foundation models represent each cell. They take the GAPDH is a gene, IL7R is a gene, COL1 is a gene. So this is one cell. This is how it takes one cell, like a book that you give to ChatGPT. What it does, it takes the most frequent genes and then says, okay, this is the topics in that book. So if you give a book to ChatGPT, it says, okay, this book is on drugs. Let's say this book is about deep sea or something like that. This book is about animals. So it will make, and then now it is trained and ready for you to use. These are called foundation models or pre-trained models. These are trained on billions of sequences, cells, and signals. What you do is zero-shot prediction. That means you give it something completely unseen, right? Like you give a completely new query to ChatGPT. It has not seen that query before, but it's still able to tell you what the answer is because it has been trained on the patterns. So similarly, these are called zero-shot predictions and foundation models. For example, today you perform a tumor biopsy, right? And now, earlier, what you would do, you would stain it and look at it under a microscope. But what you can do today is give all that data, take the data for, let's say, 1 lakh single cells, give it to a foundation model. It's an AI model like ChatGPT, which specializes on biology. And then it will give you what kinds of cells are present in it. Okay. Right. So what I've done is used a zero-shot integration model here, which is available to everybody publicly. You don't have to code. We don't have to do anything. All you have to do is run this model. It's called SCGPT. All you have to do, what I've given is data from cancer, and all it does, and then it will give you the cell types. This is from a tumor biopsy data set, and the model will run on your data real time, and it will give you this. This means that you don't have to code. All you have to do is just give the model your data and it will be able to do this. And then finally, we come, these are SCGPT is what I showed you. Then we come to AI agents. Can anybody quickly tell me if they know what AI agents are? Okay. So I'll give you a quick example. Okay. So far, for example, you want to go to a conference, right? Or an event, you want to participate in that conference. And you want to, plan the whole event. Usually what we do is we plan the event, we, and like first realize where the location is, then we see how to travel over there, then we book the dates, we book the flights or the trains, then we checking the dates, we book the flights or trains, then if you have to present something, you take that material, print it out, pack your clothes and everything, and then travel over there. The next advance that we can do right now is ask ChatGPT what's the best way, what's the best platform to get tickets, right? What is the, what are the cheapest flights available right now? And things like you can ask it to make a calendar for you, right? And all of this is what ChatGPT can do. ChatGPT can give you solutions. For example, today you want to understand, okay, I want to understand what mutations are there in lung cancers. ChatGPT can retrieve information that's already there, right? That is what it can do. But what if we go one step ahead? So one step ahead here means that if you're planning to go to a conference, there is an AI model that will take all the details, that has access to different real world tools. What it can do is not only plan your travel for you, but also book tickets for you, right? It can book tickets, it can take your calendar and mark it in your calendar. When is it? So that it can send you a reminder. It can pay for your tickets. It can book your hotel rooms. It can print your poster for you, mail it to you so that you don't forget it. That is what are AI agents. If anybody is a Marvel fan here, I think you'll know who Jarvis is. And these are like rudimentary versions of Jarvis. If Tony Stark says something, Jarvis does it. And even before Tony Stark says, sometimes it optimizes and does it even before he says it. So AI agents are also are now in vogue in the world, and they are also capable of doing things automatically. This is called autonomous computation. How are we going to apply it to science? So this is what I told you how AI agent works. AI agent has access to you, first of all. Then it has a memory, a reactivity, and a lot of real world tools. So like I said, real world tools means maybe the website of a flight, right? Where we book flights, like Skyscanner or something, where we book flights. Then it has access to your bank, it has access to your calendar, your email, and you just have to say, and it's able to do all of this for you, not just plan something but do it for you, right? So that is what an AI agent is. So till now I've spoken about how you can do your own analysis, right? So with the advent of agentic AI or AI agents, what if there is an AI model that can not only discuss with you, decide on the best experiments that you can do, but also do it for you, so that you can focus on what is more important, what kinds of problems to solve, instead of worrying about how will I get that data, how will I filter that data, how will I clean it and all. You can focus on what is more important, the problem itself. AI agents can automate entire workflows. They can get you data, says the data I showed you, right? What would humans do? You would go to that website, search for it, and then download it, and then clean it. Then you do quality control. You perform your multi-omics integration, pathway analysis. Then you generate a report. You have to write a report, right? If you perform an analysis, you have to write a report. So all of this, what if AI agents can do, and all you have to do is talk to it in human language. You don't even have to code anything. You talk to it like you talk to ChatGPT, and it's able to do complex tasks, not only in the real world. People even make AI do their stock market investments and all. That's what we do at Bolson Labs. What we are trying to do here is, let me just quickly go. Right. What we're trying to do is our agent, all you have to do is tell what you're interested in, and our agent will be able to plan customized workouts for your problem statements automatically. get data, process high dimensional data like I've showed you. This is a knowledge graph that you can see. Conduct your omics experiments and give discovery solution, not just omics experiments. It can also, so this is coming soon. The omics agent is currently under works. I'm working on it with a great group of people. It will soon be available for everybody. But what we also have currently is that other parts of drug discovery, right? Antibody design, enzyme design, bold chem, where you can do virtual screening, and retrosynthesis are also available on our platform currently. So I hope you got what AI agents are and how they can help you. Let me quickly, because this is a session on AI and biology, I have to quickly give you important caveats. So far, we've understood what a patient, how the vision of a patient changes from somebody with symptoms and test results to somebody who is a huge knowledge base of information on their genomic, epigenomic, and all different omics levels. What you can do is integrate them using different AI algorithms. Some of them are coded, some of them you don't even have to code, like foundation models that I showed you and our AI agents. What you can do out of this is disease risk prediction, disease subtyping, biomarker discovery, and molecular insights. We can also predict response to treatments via these. However, quick caveats. This is important because we are using AI. AI has a tendency, we call it garbage in, garbage out. So it depends on what quality of data you give AI. So you give good quality of data and it will give you good results. You also have to be careful of bias in these data sets. You can't just expect AI to understand what data it is, resolving. You also have to be careful because always a human expert is central in this, and then who is interpreting it and how they're interpreting the data is also necessary. However, the possibilities are limitless. And again, I will reiterate why it is important for medical professionals and clinicians to understand this is because uh, biology has shifted to omics, and now omics is shifting to medicine. Like some of you said, precision medicine. Uh, Imagine you have a complex patient and a rare disease. You earlier you would have a limited knowledge of even in the internet. Let's say you have limited knowledge of how the person would behave and how what literature is available for that disease. You don't have to do that anymore because you spend all this time studying diseases, every mechanism, every diagnosis possible. Now you can study your patient and give prescribe treatments that will actually help them. But yeah, all this said, AI cannot replace humans, not yet. But what it will do, it can be a great assistant and let omics and AI help you. That is also our motto here. What we want to build are autonomic science workflows that can help scientists, that can help clinicians, that can help uh, medical professionals to make better clinical decisions, to make precise clinical decisions and treatments. Thank you so much. You can check our website over here. Our suite, with the tools that I showed you, is already working, and you can go sign up into this and check out how it's working. That way you'll know what tools are already there, and our omics tool will be available to you soon if you want to use it for your hackathon and so on. Or if you want to just explore, you can go to the foundation model that I showed you. And this is our LinkedIn. Scan them, and yeah, you can connect. Harsh, I think, has shared how to connect to us. I'll be happy to take any questions. Thanks a lot, guys. Thanks for participating. Thanks for inviting us.
Uh, so let's wait for 1 minute if participants have any question.
Sure.
Yeah, one person asked like, I would like to know about radiomics, which was mentioned in the webinar description. Okay.
So yeah,
Yeah. So in the current webinar, we have with the time constraint, we are not able to go up to radiomics, but so that's why we have covered all these three parts. I think
yeah,
in future webinars we can go to the radiomics. I'll surely update you all. Thank you.
mm,
Yeah.
yeah. And also, radiomics is in a very, very nascent stage. But what you can look for definitely is transcriptomics, bulk and single cell, which I think will help you a lot to understand diseases better right now.
Okay. And ma'am, I have one doubt. So
Yeah,
you have
please.
said zero shot biology and one more foundational models.
Yeah.
So both are doing same work, right?
It is so foundation models are a form of zero shot biology. So zero shot biology can be done multiple ways. Foundation models are one of them. What it basically means, zero shot means there's no training over there. You don't have to train anything. It's already pre-trained, and then you can use it for a new prediction directly. I
Okay.
would have showed that as well, but a time constraint.
Okay. Okay.
But you can use this. I'll put it in the SCGPT collab, which you can use. I'll also share the link to that.
Sure.
You can try it out.
Okay.
Any other questions?
Good evening, ma'am. Am I audible?
Yeah, please tell me.
Yes, ma'am. My name is Aryan Manhas, ma'am. Ma'am, I wanted to ask, like, you've been involved in this field, and I wanted to ask, how long has AI like been a part of like your job, and where do you see it like lead to in the future? Because we obviously discussed all of this. I just wanted like your personal opinion on this, because you have like the most experience on this topic out of all of us. So I just wanted your personal opinion. Where do you see all of this going? Like, let's say from 5 years from now, even.
Right. See, I'm a biologist first, so I will always speak from that perspective because we all know that AI is heavily dependent on data. Like I told, it's garbage in, garbage out. The kind of data you give, the kind of results you will have. But even then, there's a lot of noise in biological data. It's called stochasticity, which is very important also for these kind of systems to sustain. Right. Right. So AI currently. I feel in the field right now will be a great assistant to speed up analysis. For example, COVID-19 was a great eye opener for a lot of people across different medical and biotech fields because until then, doing rapid drug discovery was just a theoretical model, not that fast as well, because we have developed vaccines and some drugs for COVID within like record times, right? Something that should have taken 10 to 11 years has been done in 2 to 3 years. And it's, although validation is a little questionable, but we've gotten to that point. So AI currently is very good for speed. Where I see it in 4 5 years is people using it extensively everywhere on every data set possible. But what is more important is to keep in mind the caveats that are there, the warnings, right? uh, but it will never be able to replace human professionals is what I feel currently, at least in the next 5 years. But it will still be a great assistant. You can speed up your analysis, you can get new insights into these and so on.
And one more thing to add, ma'am. I think most of these workflows that we are doing, as you said, analysis and also cleaning the data, I think these will also become automated. And I think most of the people will be thinking on or working on which kind of analysis can be done or which data we can integrate. Am I
Yeah.
right?
Yeah, so the what becomes more important is that what kind of problems you want to solve, what kind of problems will help people the most, right? It is very easy to have data sets and then put it into an AI model and get results. But what is more important is what kind of data sets you're choosing, what kind of problems you're trying to address, and how much of this is actually actionable. I think AI will bring us closer to that. It will rapidly increase the speed and also decrease the cost of these analysis.
Okay.
I
Thank you
see
so
it.
much, ma'am. And one last question before I understand the time constraint, but one last question, ma'am. I
Yeah,
wanted
go
to
ahead.
ask, like, I'm a first year MBBS student, ma'am. So I
Okay.
wanted to ask, like, if I want to learn more about this field, get more knowledge about omics in general.
Okay.
So what advice would you give, like, for a first year medical student?
Yeah, great. I would really encourage all medical students to, explore omics, different layers and everything. What you can do for starters is try and understand different omics layers. I think ChatGPT, Claude, and other models will be a great help in understanding these. What you can do is kind of understand how these kinds of data sets are used for prediction and so on. So this foundation model that I shared is one way. I can also share a set of resources through Harsha and Pavan later for you guys to, get started with omics.
That
So you
would be
can start with
amazing,
foundation.
ma'am.
Yeah, and also I would really advise you all. Like I started as a biologist, right? So do not be afraid of coding or code. Do not be hesitant to try out code. Uh, don't get afraid seeing code and try these foundational models, and I think you will see how amazing they are.
Thank you so
I see
much,
a
ma'am.
You're welcome. I see a question in the chat box. I'll quickly answer that and we can wrap up. Are the
Yes, ma'am.
results
We'll
that you
take this
guys are
last question.
yeah, results that you guys are working on with foundation models not yet framed? How can we ensure that we ask those questions, making sure that we utilize our time? So we are not working with foundation models, we're working with AI agents. Foundation models, you'll still have to give the data to the model and run it. But AI agents, you just have to give a prompt like you give to ChatGPT and they'll perform the entire analysis. So we are still testing to see if they're answering questions we have not yet framed. I'm not sure what that means, but if you give a question that does not have an answer yet, for example, how to treat a certain genetic disorder, right? The AI will try with its best ability and the knowledge that it has to come up with a solution, but that has to be validated by a human expert. It may or may not be right. Obviously, we have not reached until there. But we can repurpose existing drugs. We can find alternative treatments to existing diseases and so on. Thank you.
Okay. So thank you so much, man. And Dr. Gayatri and Mr. Harsha, thank you so much. You have very well explained omics. Like you have removed the whole jargon about omics. Like many people think it's very complex. You have simplified it very well, and also have told like how we can take this data and make and use it in a way in which it can be helpful for the people and healthcare. And also, the next session will be on the next week or other week. So it is on AI Boltzmann suit. So now ma'am has showed at last quickly about their platform. So we'll be going detailed into their platform in which they will be explaining about how to do small molecule discovery and protein engineering. So please do join. I'll circulate it. And also one more thing before concluding. Uh, I would like to just inform you about the Nobel Research Sprint event through which we are conducting this webinar. So just give me a minute. Yeah, I think my screen is visible right now.
Sorry
Yes,
to interrupt you, Pavan. I think I have to leave right now.
Yeah.
But thanks a lot. I hope the session was helpful to you guys, and Harsha will be in contact with you.
Okay, sure, man. Thank you so much. And
Thanks. Thanks.
I'll just wrap it up in 1 minute. I'm just going through the event. So this is the event Nobel Research Sprint.
Sure.
This is our website. I'll put the link in the chat box. You can go through it of our website. So this event is like an ideathon in which you should form teams with the different disciplines like biotechnology, bioinformatics, or physics, chemistry, engineering. Any people like come up with four team of 4 members and go through the problem statements, and now you can use this knowledge of omics to giving your solutions or ideas. So this is our website. You can go through it, and here is timeline. Basically we have December 25th as submit last day of submissions and December 15th as last day for registration. And here are our mentors from IASC and AIMS New Delhi. So this is our registration details. You can go through it. I'll just attach it in chat box. Hello. Yeah. Okay. So is the link available in the chat box? Are you guys seeing?
no,
One.
sir.
Just give me a minute. I'm just adding. Yeah, I think you will find now. Yeah. Mm, yeah. I think now you'll get. So please copy that link. I has sent it in the chat box. And please come with your brilliant ideas and make teams. Okay.
Thank you so much, sir.
Okay. Okay, then see you again in the next webinar. Bye, everyone. Mr. Harsha.
Yes, Pawan.
Ah, so like now we can wrap the webinar.
Yes, yes.
Okay.
Thank you everyone for joining in. If you have any queries, you can reach out to us to the email ID that we mentioned, or else you can reach out Pawan. He will redirect you to us.
Okay.
And wish you all the best for this hackathon.
Okay. Okay then. Okay. Bye, everyone. Have a nice day.